I really enjoyed this morning's session, as is fairly predictable at these sorts of events. Some of the things that I was going to say have already been covered. But when I was putting together my talk, I was fairly sure you wouldn't have got my first slide. So this is my puppy Joe, and I thought I would lead with him because Joe's a wolf, right? You can see he's clearly a wolf. He's got the legs, the ears. He's a wolf. And when he sees a deer, he knows he's a wolf. He's going to have that deer. He's only this big, right? He's a small dog. The reason I got Joe up is because my ancestors have selected his ancestors for traits that they observed and they wanted to propagate. And so Joe clearly isn't a wolf. He doesn't look like a wolf. He doesn't behave like a wolf other than when he sees a deer. And you can look at the genetic signatures of that selection. You can see changes in oxytocin signaling pathways because Joe loves a cuddle. And you can see changes in carbohydrate metabolism because he can and will try and eat anything that I do. But obviously, we haven't just selected wolves. We've applied this same process of selection for observable traits throughout recorded history. So all of our livestock species have been selected and have changed from the wild type variants from which they're derived. Now, in recent years, we've heard quite a lot this morning about genomic selection. So this is my very layman's view of what's involved in genomic selection, because I don't do this. So we take lots of animals, identify thousands of SNPs, associate those with thousands of phenotypes, and generate a prediction equation. You then take another population of animals, you collect the SNP profiles, you don't phenotype the animals, you just apply the prediction equation and generated an estimated breeding value. You use this estimated breeding value to select the animals that you want to breed, depending on what parameters you're looking for, and you produce a better animal. And this is an iterative process. So there's this animal and his siblings go back into this population. You apply the estimated breeding value again, select your animals, and over generations, you get better and better animals, okay? Now, this process clearly works very well. It's a very powerful tool, not least because you don't need to understand anything about the genetics that's underlying what it is that you're doing. You're selecting for a trait based on what the computer says. Computer says, do this, so you do this. I'm not putting this down, don't get me wrong. This is brilliant. It works very, very well. It does have one major limitation, though. It might have more. It has one that I've identified. And that is you're limited in your selection parameters by the genetics of this population over here. If the trait that you want isn't encoded in the genome of the population you're selecting for, there's not a lot you can do about it other than look elsewhere. So let's say we found a trait for blue ears and a blue nose. And the breeding companies, in their wisdom, decided blue ears and blue nose was a trait they really, really wanted. But this animal has not been selected. It's very difficult to bring this into this population without diluting your hard fought for gains that you've managed to achieve over generations of iterative selection. But using biotechnology, obviously, if we understand the genetics of blue ears and blue nose, we can put it straight into the population, into the elite animals. Now, we are not the developers of this technology, but by biotechnology, I'm meaning anything in the suite of tools that we can use to directly manipulate the DNA of the animal in situ. So we have a lot of options available to us. We can do anything from transgenesis, where we put an expression cassette in, so this is a promoter and a transgene, through to changing a single nucleotide using gene editing. And what we have actually is a spectrum of things in between. And I find it difficult sometimes to determine where editing ends and transgenesis begins. I think largely the regulators are struggling slightly with this as well. Now, as I said, we are not the developers of these tools. Other people make them for us. We just use them. And the reason these tools are being developed is largely not for our benefit. So I'm going to talk very briefly about transgenesis. I know most of what we're talking about here is going to be down at this end of the scale in the edited, but actually I just want to do a couple of slides on transgenesis first, because when I first came into this field, I went to a talk at the UC Davis conference in Tahoe, and there was a group from Guelph University who had produced the EnviroPig. And I thought, wow, that is something that is really useful. That is a really good use of technology. And so in pigs, when you feed them phosphate in their diet, they get organic phosphate in their food. Organic phosphate is not very digestible. So most of the phosphate that goes in comes back out again. And this is an environmental contaminant. And what the guys at Guelph did was they took an E. coli phytase gene and put it under a parotid secretory protein. So it's produced in the salivary gland of the pig, such that the pig can convert the organic phosphate into inorganic phosphate, which is very readily absorbable. So far less phosphate coming out at the other end, far reduced environmental impact of the pig farming. Now, the timing of this probably wasn't very good. It was an excellent study, and I thought, brilliant, but transgenesis is not well received and has not been well received. The Aqua Advantage salmon took, I think, 26 years to get approved. And you have to have very deep pockets to go through that process. So this particular project didn't go anywhere, but the science is still progressing. So this is a recent paper from a group in China. This is phytase. There's also a couple of glucanases and xylanase in there, which break down very complex sugars in the food. And what you can see, the bottom line is, this is the amount of stuff in the feces, the nitrogen, the phosphate, and the calcium. The transgenic are the black bars, and the wild types are the spotty bars. So I think we're missing a trick still here by moving entirely towards editing. I think transgenesis still has a lot to offer. But I don't do very much transgenesis. I do editing because we can see a pathway to market with editing. So editing is not transgenesis. Editing is using a suite of tools, so zinc fingers, talons, the CRISPR-Cas system, to identify a sequence of DNA and create a break at that site. In essence, you can think of it as the edit find function in Word, and then you hit the shift button to create a line break, and then you manipulate what happens at that breaking and let the cell fix it itself, and you get non-homologous end joining events, and that's great for just breaking a gene, turning it off. But you can supply small templates as well that will lead to specific point mutations that you're looking for at the target site, or you can use it for integrating a transgene of interest at a specific site in the genome. Now, the words that I came out with before, we are not the developers of this technology. I've got my slides right in my head again now, right? We don't develop this technology, other people develop it for us, and they're developing it largely for biomedicine, right? These tools are used for biomedicine because there's a lot of money to be made in treating human disease. And there are a variety of human diseases, this is not an all-exclusive list, that are either being investigated or have started to be successfully treated using genome editing. Now, in biomedicine, when applying genome editors for these diseases, you're looking to make a change in a specific tissue. What you're not allowed to do is change this, right? Germline genome editing, there is a moratorium in human germline genome editing. But of course, for livestock, we want to do it the other way around. I have no particular interest in changing a gene in the muscle of an individual pig. I want to change the gene here, right? We want to change the gene in the single cell zygote most often, because by the time you get to this little thing here, the germ cell niche is already occupied. right? We are just carriers for our DNA for the next generation. So each of the species, each of the groups of species that we are looking at have different constraints, different challenges in editing that single cell zygote. So I'm going to go through these in sequence. I'm going to start with this. So this is a mammalian blastocyst. I think this is a sheep, I want to say. I took the photograph, but I can't quite remember. It's either sheep or pig. And so once you've got this, it's relatively easy to genetically manipulate it. You can use a variety of techniques. Our limitation actually with this is not the manipulation of this, it's getting enough of them, because it's quite expensive, for me certainly, to get large numbers of oocytes, which we then do IVM, IVF, get our zygotes. But the major constraint actually is you can't grow this in an incubator. You need a surrogate. So you're talking large numbers of animals to actually incubate your manipulated embryos. And there's no way around that at the moment, but we might get there at some point. So when we're talking about genetically manipulating the single cell zygote, we have a number of options. We can use cloning. So we can take cells from this animal, we can culture fibroblasts, we can take an oocyte and we can enucleate it, electrofuse these two, produce a blastocyst, transfer it into a recipient and establish a pregnancy such that you produce a lamb which is genetically identical to this, right? And because we've got cells in culture, we can genetically manipulate the donor cell such that the cloned animal is genetically identical to this with the addition of the genetic changes that you've made in the single cell. Cloning works reasonably well, but the efficiencies are not great. You sometimes get large offspring syndrome, presumably due to epigenetics of the culturing at this stage. It works pretty well in pigs just because of the numbers you can throw at it. In other species, it's actually quite expensive to do cloning, but we have other alternatives available to us. So this is what I do, just a little needle, micro-inject whatever the reagents are that we're using directly into the zygote. You can use AAV vectors, so these are non-integrating viruses to deliver to the zygote. You can electroporate to deliver to the zygote. So the actual genetic manipulation of this is not a problem. In fish, we have different constraints. So we've heard a little bit about fish today. You have almost unlimited access to eggs and sperm because fish produce a lot of them. You don't need a surrogate to host them. So you just, as long as you've got good culture, aquaculture conditions, you can grow them up. This is my artistic representation of a salmon egg. And so you've got a big yolky structure with a single cell sitting on top. And so genetic manipulation of that is relatively straightforward if this is the size of the thing that you're working with. They're actually a bit smaller than this, right? So what you're aiming to do is to inject into here, but because of the scale, and I've tried this, I'm not very good at it. It's very easy to go in a little bit too high or a little bit too low. This is a very skilled procedure, but it does work, and people who are good at it do it very well. And so we have modified salmonids, we have catfish, tilapia. Biorender didn't have a pufferfish, so I drew that. There's red sea bream as well. So there are a variety of species now that have been successfully genome edited in fish. So the technology does work extremely well in this sphere. Chickens are different again. So we have pretty much unlimited access to fertilized eggs, right? When it's ovulated, what is ovulated is this yolky mass here with the single cell sitting on top of it. But it then takes 24 hours to pass down the oviduct. You get egg white secreted around it. You then get the shell. It's then laid. At point of lay, this single cell is no longer a single cell. This is a sheet of about 50,000 cells. So accessing the single cell in a laid egg is virtually impossible, getting into that germline with genome editors. But in chickens. So this is an embryo about 3 days. And at this point, the primordial germ cells, which were resident in the germinal crescent up here, have migrated into the bloodstream, and they're heading towards the primitive gonad to populate it. And what you can do in chickens is take a small amount of blood out of the bloodstream at this stage, and you can culture out the primordial germ cells. And again, because you have cells in culture, you have an opportunity to genetically manipulate those. So these are single cells that will eventually contribute to the germline. And once you've genetically manipulated these, you can inject them back into the bloodstream of an age-matched recipient. So this is a picture of Maeve Ballantyne. She was in Mike McGrew's group at Roslin. And in her hands, she has 2 chickens. This one on the right, as you're looking at it, has the dominant white phenotype. So this is actually a mutation in a feather color gene that means they lose their pigmentation. So all of the white chickens you see have dominant white phenotype. And what Mike's group did was to genetically modify, to edit the PGCs to restore the feather color function in this one here. So this works incredibly well. And this is a really powerful technology because you have access to both sexes at the germline stage. Depending on the sex that you take it from, you have either male or female. You can modify them. You can put them individually back into recipients that in one generation you can cross to get homozygous animals. Now, in mammals, we don't have this available to us at the moment, but what we do have is the spermatogonial stem cell isolation culture and the surrogate sires technique that John's going to talk about later today. So using this, you can take a testicular biopsy, you can dissociate these cells in culture, you can use a multi-parameter selection to culture and expand the spermatogonial stem cells, which you can introduce back into the testes of a recipient. And again, because you have cells in culture, we have an opportunity to edit those cells. Now, this does only give you access to the male germline, but what it means is that the offspring that you're going to get out will be genetically related to this animal, not this animal. And if we've edited the cells, it'll be edited as well. And John's also shown that not only does this work in pigs, but it also works in cattle and goats. Now, as I said, this is only one side of the gene editing story because we can only do this in boys at the moment. But just over the horizon in rodents, what's been shown is that you can take stem cells. So these can be embryonic stem cells or induced pluripotent stem cells. And using culture conditions and growth factors, you can differentiate these into primordial germ cell-like cells. And you can use those to repopulate the testes of boys and girls and produce viable eggs and sperm. My picture is a little bit misleading because I don't think they've taken this and added them together to make this, but they've used this with wild type eggs and they've used this with wild type sperm and shown that you get viable offspring produced using this process. Now, we're not there with livestock at the moment, but I think it's entirely possible within the next years that we might be. And I'm just going to finish with a challenges slide. So the technology works. The technology works well for us at the moment. It's not perfect. It is improving. One of the things that's come up in conversation earlier today is public perception. It's important to recognize there's no such thing as the public. I think Alison said that once. I think I nicked that. But everyone has their own views, right? Depending on your own life history, your friends' views, your parents' views, what you see on the television, you all have your own views. And it's incredibly polarizing. And I think what we need to do is identify the middle ground and then build on that rather than starting from poles and butting heads in the middle. The efficiencies of the tools we have at the moment, I said, they're fine for me for doing science. They're actually probably somewhat challenging for large companies because what you're needing is to make the same edit again and again and again reliably. And at the moment, I don't think we're there with respect to the editing technologies that we have. We're actually short of targets, right? We have lots of things that we would like to be able to do, but we don't know what to edit to achieve that. Causative SNPs that have been identified are rare. And actually, it's likely that most of the production targets that have been mentioned earlier today are going to be polygenic. And again, the efficiency of the tools at the moment are not ideal for polygenic traits. And finally, just regulations. It's why we're here to talk about regulations today. It is problematic because they vary between jurisdictions, and we really need some form of harmonization so we don't end up with trade barriers when we implement these projects. And I think that was all I was going to say. Ta-da!